We're starting our second part of Chapter 5, Microbial Metabolism. And the only objective for this section of notes is that you should be able to identify different factors that influence enzyme action. We talked about different characteristics of enzymes in our last PowerPoint. So there are two types of cellular controls on enzymes: control of enzyme synthesis and control of enzyme activity. How active the enzyme is, or how much enzyme is present. And so the main influencing factors that affect the activity of an enzyme is temperature, pH, substrate concentration, and presence or absence of inhibitors. So we're going to look at each of those factors. So when we look at temperature, rates of chemical reactions increase as temperature increases. So as we heat up things, and you think about that even in the lab, as you heat up things either with a Bunsen burner or a hot plate, you're starting to increase those chemical reactions that can occur. Same thing can happen in an organism. As the body temperature increases, so do those chemical reactions. But if you elevate beyond the optimal temperature, that actually reduces the rate of a reaction. Okay, so when you think of in here, we have our body temperatures right around 35 to 37 degrees Celsius. So if we start going too much past that optimal temperature, then we're going to start to see denaturation. Okay. Loss of that three-dimensional structure of the proteins. So the proteins denature. That's what we call this process right here. Breaks hydrogen bonds and non-covalent bonds, change the shape of the activity site, of the active site, so the enzyme loses its function. Remember, the substrate can only connect to that active site because it's a very specific shape for that specific substrate. So if all of a sudden we start changing that shape, now that enzyme isn't going to function anymore because the substrate can't hook onto it. Sometimes this can be only partially an effect, or it could be fully reversible, perhaps, if we were to bring back the temperature back down. But other things besides temperature, acids, bases, alcohol, and UV radiation can also cause denaturation to occur in the proteins of the enzyme. So let's just take a look at what this might look like on a graph as we approach optimal temperature right here. So looking right between 35 and 40 degrees Celsius right here. Optimal temperature, that's when you have the highest rate of enzyme activity right here, okay? But as soon as we get too far beyond that, if it starts heating up higher and higher, look at how fast that drops. With pH, most enzymes also have an optimum pH. So if you get too acidic or too basic, that can cause denaturation as well. So here you have a protein that's all folded and coiled up how it's supposed to be with its protein folding. And when it gets exposed to an acid or base, it might start to unravel and denature, and pretty soon it doesn't have its shape that it's supposed to have. If it doesn't have its shape, the substrate cannot attach to that active site. So again, if you were to look at that in a graph form, right up here, it looks like this one, about a pH of 5, is about its optimal pH right here. But if it gets too much more acidic, or starts moving into more of the basic area there, it loses its function. The enzyme doesn't function as well. Substrate concentration just deals with the amount of enzyme. There's a maximum rate at which the amount of enzyme can catalyze a specific reaction. Okay? When the substrate concentration is extremely high, we say that the enzyme is in a state of saturation. Okay? That means that every active site is always occupied by a substrate. Okay? If you increase the substrate, it will not affect the rate because all the active sites are already in use. Okay? It's kind of like, if you're going to go on a ride at the fair, once all of the seats are taken up on the ride, you can't add more people to it because they're already filled. So same thing with an enzyme. If all the active sites on the enzymes are already full, in other words, they are saturated, it doesn't matter how much more substrate you add to the reaction, you're not going to get any more of a reaction rate happening because, the enzyme, they're full. You've got as many as you can do right there. So usually enzymes are not saturated with substrates under normal cellular conditions. So if you suddenly try to up the substrate concentration, you can influence the rate of the reaction, okay? Because normally enzymes are not saturated under normal conditions. So if you look here, you can see that if you keep adding more and more substrates, enzyme activity will increase until, all of them are full. But from this point on, it doesn't matter if you keep adding more substrates. It's still a flat line. It doesn't keep going up because, too bad, all the enzymes are filled. All their active sites are filled. So it's just a flat line like that. Now this last topic of things that can kind of affect how enzymes work deal with inhibitors, okay? And there's two types that I want you to know about. There's what we call competitive inhibitors and non-competitive inhibitors. Competitive inhibitors fill the active site of the enzyme, and so they compete with the substrate. That's why they're called competitive inhibitors, okay? So let's just say there's a certain seat that you always sit at school. Let's say like you have a certain desk that you sit in for a certain class, and somebody else all of a sudden came in and sat in your desk for that particular class, and it's like, well, where are you supposed to go then? That's your seat, right? They competed for you for that spot, and now you can't sit there anymore. Okay, so if this was a particular substrate that needed to hook onto that active site so a certain reaction could take place, either a breaking down of a molecule or joining together of two smaller okay, now this can't occur anymore because this competitor has come in and taken the spot. These can be reversible or sometimes irreversible. Sometimes if you increase the substrate, it might overcome the competitive inhibition. You just might, if you overwhelm it with more and more substrate, it's kind of like they can crowd out and take out that competitor and take the spot instead. Non-competitive inhibitors do not compete with the substrate for the active site. What they do is they interact with what's called the allosteric site. Okay, this is another region on the enzyme, and these inhibitors hook onto there, and it causes the active site to change shape. Okay, remember, that active site has a very specific shape for a very specific substrate. So if you now change the shape of that site, it could make it non-functional. Now the substrate can't hook onto it because now you've totally modified the shape of it. So that substrate will come along and it's like, oh, I can't hook onto this because I don't have the right shape for this. So this can also be reversible or irreversible. Sometimes allosteric interactions can activate an enzyme rather than inhibit it too. So sometimes it can work the other way. Can operate on enzymes that require metal ions for activity. So some things such as poisons such as cyanide and fluoride can permanently inactivate enzymes by doing this. So let's take a look at what this looks like. So for competitive inhibition, here's the substrate. It has a very specific shape. You can see this, and it hooks onto this active site. Well, if we have an inhibitor come along and block the site, now look at this cannot fit in there because this portion of it will not be able to hook onto that. And so that competitive inhibitor has now inhibited or stopped this substrate from being able to hook onto this enzyme. Non-competitive inhibition is when we have this area right here called the allosteric site. You can see it right here where it's labeled. Okay, so this non-competitive inhibitor hooks on there. Nothing happens up here. Nothing hooks on here. But notice what happens to the shape. As soon as this non-competitive inhibitor hooks on here, this turns into kind of a funky shape here. Now this doesn't fit anymore. And so it's gonna not hook on anymore. So it's inhibited it from working, that substrate from hooking on, not by blocking its site, but by changing the shape into this weird thing here that's not even the same shape as this right here. Also, we have what's known as feedback inhibition. And sometimes what feedback inhibition does is it's end product inhibition. Basically, it stops the cell from making more of a substance than it needs and wasting chemical resources. Okay? So it's basically an assembly line where each step has a specific enzyme, and the final product may inhibit the activity of one of the earlier enzymes. So if you all of a sudden have a buildup of the end product, it may actually go and block the enzyme's allosteric site until we've used up a lot of that end product, and then the pathway can continue. So if you look at that, here we have your substrate, it hooks on, and the enzyme is there, it makes its product, it goes just fine, and it produces your end product right here. But if all of a sudden this builds up, now all of a sudden, you notice this allosteric site? Now all of a sudden this end product binds to the allosteric site, and it basically shuts down this whole pathway so that no more of this end product gets produced until we use up some of this end product. Because why would your body and your cells keep producing more and more and more of an end product if we don't need it? Whether that be a protein or some other substance that we're making, that would be just a waste of our cells, functioning and ability. So feedback inhibition is a way of kind of temporarily shutting down our enzyme and just saying, okay, we've got enough of this end product, let's just wait till we use it up. And once we've used it up, guess what? Once we use it up, this will be taken away from here. And now we have an empty spot here again, and so we can produce more of that right here, okay? So feedback inhibition is also a way that can kind of controls that. And then we have ribozymes. Those are unique RNA that function as a catalyst that bind to substrates. They're not used up in reactions. They specifically act on RNA. So when we talk about transcription, once the messenger RNA copies the code from DNA before it goes to a ribosome to be made, translated into a protein, messenger RNA gets processed a little bit, where it has some of the introns get removed and the exons get spliced together. Well, these ribozymes are going to help in that process there of doing that. And that's the end of this section.